Good morning, everyone. Today we are going to dive into a fascinating topic, the functions of cytoskeleton in plant cells. Now, when you hear the word cytoskeleton, you might immediately think of structure, and yes, that's a big part of it. But in plant cells, the cytoskeleton isn't just a static framework, it's a dynamic, adaptable, and crucial for the process like transport, cell division, and even how plants respond to their environment. Throughout this lecture, we will explore how cytoskeleton contributes to plant cell organization and survival, and we will see how its components, microtubules, and acting filaments work together to achieve some truly remarkable feats. So let's get started. Introduction to the cytoskeleton. Before we dive into the specific functions of cytoskeleton in plant cells, let's make a moment to understand what cytoskeleton is. Cytoskeleton is a highly dynamic and complex network of protein filaments found within cytoplasm of cell. Think of it as a cell internal skeleton, but unlike a rigid framework, it is constantly recognizing itself. This makes it incredibly versatile, also enabling the cell to adapt to various needs. In plant cells, cytoskeleton plays a critical role in maintaining cell shape, supporting intercellular transport, organizing organelles, and driving processes like cell division. Without the cytoskeleton, the integrate internal structure of plant cells would collapse, and essential functions like movement of vesicles or positioning of chloroplast wouldn't occur. So it's important to note that cytoskeleton isn't just a structure, it's a backbone of dynamic processes that keep cell functioning and growing. So let's now break down further it into its key components and their specific roles. Move on to the next slide. Now that we have set the stage, let's drive into the cytoskeleton itself. So cytoskeleton is highly dynamic network of protein filaments that exists throughout the cytoplasm of plant cell. Think of it as cells internal scaffolding, providing structural support while also enabling movement, transport, and communication. In plant cells, cytoskeleton plays a dual role. It not only gives cells shape and stability, but also coordinates essential processes like intercellular transport, cell wall organization, and cell division. Unlike in animal cells, in which whereas plant cells are surrounded by a rigid cell wall, this means cytoskeleton has to work in harmony with the wall to control growth, polarity, and overall cellular structure. The cytoskeleton is composed of three major components. First is microtubule. Microtubules nothing but hollow tubes. Also, you can see in the diagram. So, microtubules are nothing but hollow tubes that control cell shape, intercellular transport, and division. And the second component is acting filaments. Acting filaments, they are thin flexible filaments involved in movement, transport, or signaling. And then there is also third one is intermediate filaments. Although they are less prominent in plant cell, but they provide mechanical support in some cases. So together these three components, they create an integrated system that drives both structure and function in cell. So now let's take a closer look at each of these component in details. Move on to the next slide. Microtubules in plant cells. So let's focus on one of the key component of cytoskeleton, microtubules. Microtubules are found in cytoplasm of all types of eukaryotic cells with rare absence, such as in human erythrocytes. Means they are not present in human erythrocytes, but they are present in eukaryotic organisms. They are tiny, hollow, bead-like tubular structure that help cells maintain their shape. They are microscopic hollow tubes found inside the cell that also provide motor functions to the cell. Cytoskeleton of eukaryotic cells made up of proteins that gives shape to it. The three types of protein that help in the organization of cells are microtubules, intermediate filaments, and microfilaments. So microtubules is present in all eukaryotic cells except human erythrocytes. It is present in the cytoplasm freely, or it can be found forming in the parts of centrioles, cilia, flagella as well. In both animal and plant cell, microtubules are present in the cytoplasm of the following sites like cilia, flagella, centrioles, basal body, nerve processes, mitotic apparatus. This is also found in mitotic apparatus, cortex of meristematic plant cells, elongating cells, selected structures in protozoa. In protozoa, they are present in it. Yes. Uh, Also, there is also a variation in stability of microtubules of various sites. Cytoplasmic in cytoplasmic and the spindle microtubules are liable. Microtubules at cilia and flagella are resistant to different treatments as well. Move on to the next slide, the structure of microtubules. So let's now take a closer look at the structure of microtubules, which underpins their diverse role in plant cell. So microtubules are composed of protein subunits known as tubulin. These subunits form dimers of two types. First is alpha tubulin and then another is beta tubulin. So these dimers stack together form a hollow cylindrical tube. The microtubule has polarized structure with two distinct ends. First one is the plus end, another one is the minus end. The plus end, where growth occurs more rapidly through the addition of tumor dimers, and the minus end, which is usually anchored. As you can see, this is the plus end. This is what I am talking about, the plus end and the minus end. The minus end, which is usually anchored and grows more slowly. This structure polarity is essential for the directionally of intracellular transport, as motor proteins like casein and dynein move along microtubules in a specific direction. Microtubules also exhibit a property known as dynamic instability. This means they are constantly growing and shrinking by polymerization and depolymerization of tubulin subunits. This dynamic nature allows microtubules to adapt quickly to the changing needs of cells, whether it's during cell division, transport, or reorganization in response to environmental stimuli. The hollow bead like structure provides strength and flexibility, making microtubules an ideal framework for tasks ranging from intercellular transport to shaping the cell walls. So, in summary, the structural properties of microtubules: tubulin dimers, polarity, dynamic instability are what make them so versatile and indispensable to plant cells. Move on to the next slide, which is about the function of microtubules. So the functions of microtubules in eukaryotic cells, microtubules perform different functions that are mechanical functions. The first is mechanical function. The different orientation of microtubules and their distribution is related to the shape of the cells, the cell processes or proturbulence like axons and dendrils of neurons, microvilli, etc. The another is morphogenesis. Microtubules helps in determining the shape of developing cells which occurs during the process of cell division. For example, during spermatogenesis, the nucleus of spermatid gets elongated, so microtubules accompany it. Similarly, during the induction of lens placode in eyes, there is also a elongation in the cells. Numerous microtubules accompany it too. The third one is cellular polarity and motility. The intrinsic polarity of some cells also determined by microtubules. Similarly, the directional gliding of the cultured cells also dependent on it. The fourth is contraction. They help in contraction of the spindle. Uh, it aids it aids in the movements of chromosomes and centrioles. They also help in ciliary and flagellar motion. The next function is circulation and transport. For the transportation of macromolecules, granules, and vesicles within cells, microtubules gets involved in it. For example, uh, uh, the cytoplasmic particles migrate in and out in the pseudopodia of actinospherium, which is a protozoan. In this long and thin pseudopodia, there is a presence of microtubules of about 500 micrometer. Move on to the next slide, which is what is actin filaments. First of all, actin filaments, which is also known as microfilaments, are dynamic structures that are made up of actin protein subunits. They are one of the three major components of cytoskeleton elements, along with microtubules and intermediate filaments. They are required for maintaining cell shape, supporting cellular structure or organelles, allowing cell mobility, and helping cellular functions such as transport. Actin filaments are dynamic with continuous polymerization and depolymerization of the actin. They are not fixed. It is influenced by the involvement of actin binding proteins that controls filament construction, disassembly leading to stability. They serve as the base of motor proteins like myosin, which allows cellular movement such as contraction of muscles, cell migration, and transportation of intercellular protein. Now talk of structure of actin filaments. Actin filaments consist of globular actin, which is G-actin subunits that polymerize into long filaments that are parallel and helical. This polymerized structure, as you can also see in the diagram, this polymerized structure is known as filamentous actin, which is also pronounced as F-actin. The filament has repeating subunits which are of 37 nanometer size, and each G acting subunit is 5.5 nanometer in diagram. This polymerization of G actin into F actin results in polarized structure with distinct barbed and pointed ends. These ends elongate or shortened by addition or removal of G-actin subunits at either in the end of the filament. Move on to the next slide. Uh, the actin, some of the key structural properties of actin are as follows. So first is compositions of subunits. So G-actin is the subunit that polymerize into F-actin that I have told you earlier as well while I was explaining the diagram. Second is polarity. They are polar in nature with separate barbed, positive and pointed or pointed, which is also known as negative ends. The third property is helical structure. The arrangement of G-actin subunits causes actin filaments to form helix. The fourth property is binding sites. They have binding sites for several regulatory proteins affecting their stability and dynamics. Move on to the next slide, which is dynamics of actin filaments. The dynamic nature of actin filaments is crucial for their cellular functions. Some of the important aspects includes polymerization and depolymerization. So actin filaments, what they do in this, they undergo constant making and breaking G-actin protein, G-actin polymerized onto the barbed end and depolymerized from the pointed or negative end. The second is regulation by acting binding proteins. Various actin binding proteins modulate assembly of filament dis, uh, disassembly and stability, thereby influencing various cellular processes. The third one is cytoskeletal arrangement. They undergo arrangement in response to extracellular signals, altering cell shape, movement, and function. The fourth one is cross-linking of the filaments. The proteins such as coffulin and fibrin regulates filament severing and crossing linking. This impacts filament organization and dynamics. Move on to the next slide. Cytoskeleton and intercellular transport. So this slide focus on the role of cytoskeleton in intercellular transport process, highlighting how it facilitates the movement of various cellular components. So here is an explanation for each part of the slide. First is a role in vesicle trafficking, the movement of Golgi derived vesicles for the cell wall synthesis. The cytoskeleton is, we all know, is essential for the transporting vesicles that originate from Golgi apparatus. There is a vesicle carry material necessary for the cell wall synthesis such as cellulose, hemicellulose or pectin to the plasma membrane. The vesicle move along the cytoskeleton networks, particularly microtubules and actin filament, ensuring that materials are correctly delivered to the appropriate location of cell wall construction. The second is the transport of organelles. So chloroplast positioning under light conditions. The cytoskeleton also helps. This point talks about the cytoskeleton that also helps in position organelles like chloroplast within cell. So under light conditions, chloroplasts need to move optimize light absorption for this photosynthesis. Microtubules and actin filaments play a role in dynamic positioning of chloroplasts within plant cells. These organelles may rearrange themselves to align with light sources, maximizing their exposure to light for efficient photosynthesis. The third is interaction with motor proteins. Motor proteins What they do, they interact with cytoskeleton to facilitate intercellular transport. These motor proteins walk along cytoskeleton tracks, microtubules or acting filaments using energy from ATP hydrolysis. Uh, the fourth point is dynein and casein for microtubules. So ki- kinesins generally move in plus n direction towards the cell membrane or periphery, and they are important in process like vesicle trafficking and organelle positioning, whereas dyneins in contrast typically move in the muscle end direction towards the cell center, usually microtubule organizing center or centrosome. They are involved in process such as endocytosis, cilia beating, and positioning the Golgi apparatus. The fifth point is myosin for acting filaments. Myosin is a motor protein associated with acting filaments, typically moving towards the plus end of acting filaments towards the cell membrane. Myosin is involved in process such as cell division, vesicle transport, and muscle contraction in muscle cell. In plant cell, myosin can be involved in vesicle trafficking and organelle movement along actin filaments. Role in cell division. Cytoskeleton plays a critical role in cell division in plant cell, uh, and the explanation of each stages are interphase and prophase band, spindle formation, and phragmoplast. We will study those stages into detail with the help of a diagram or image. So let's get started. The first component is uh, interface, in which cortical microtubules involved. So cortical microtubules regulate the direction of cell elongation, as you can see in the diagram as well here, and here is a microscopic image. So cortical microtubules regulate the direction of cell elongation by guiding the deposition of cellulose microfibrils in the cell wall. They also help together organelles and facilitate intercellular transport. So the arrangement of microtubules during this phase ensures that the cell maintains its shape and prepare for the division process. And in second stage, which is prophase band, pre-prophase band, which is PPB, the pre-prophase band, a ring of microtubules and actin filaments forms during G2 phase of cell cycle. It determines the future plan of cell division, providing spatial clues for mitotic spindle and cell plate alignment. In case of unequal division, as seen is asymmetrical division, the PPB establishing the axis of division. The disappearance of pre-prophase band before metaphase doesn't disrupt its positional cue for division. Third stage is spindle formation. So microtubules in this stage, microtubules reorganize to form mitotic spindles, which is essential for separating sister chromatids. The spindle apparatus ensure accurate chromosome segregation, preventing genetic errors in the daughter cells. Spindle microtubules attach to the chromosome via kinetochores and apply forces to chromatids towards the opposite poles. The fourth stage, which is phragmoplast. Phragmoplast is a microtubule and acting filament based structure that forms between daughter nuclei during cytokinesis. So it guides the assembly of cell plate, a structure that eventually develop into a new cell wall separating two daughter cells. The phragmoplast directs vesicles carrying cell wall materials to the division plate, ensuring proper cell wall formation. The next slide, uh, the role of cytoskeleton in in mediating environmental responses in cell, focusing on how it participates in signal transduction, mechanical stress responses, and the growth directionally. Here is a breakdown. First one is role in signal transduction, transmitting signals from environmental stimuli. Cytoskeleton act as scaffold for transmitting and amplifying signals from cell environment. When cell encounter external signals, for example light, gravity, temperature, or chemical signals, the cytoskeleton facilitates intercellular transmission of these signals to trigger specific response. Cytoskeletal components like actin filament, microtubules can undergo dynamic reorganization in response to external clues, helping cell adapt to changes in its environment. For example, in plant cells, the cytoskeleton plays role in mediating responses to pathogen attacks or changes in water availability by altering its structure to force cell walls to redistribute organelles. The second is response to mechanical stress. The recognition of cytoskeletal elements, the cell experiences mechanical stress from various sources such as pressure, tension, shear forces. Cytoskeleton is crucial for sensing and responding to these forces. Microtubules and acting filaments dynamically reorganize to reinforce areas of stress, redistribute forces, and maintain cellular integrity. The third is photoperiodism and gravitropism. Cytoskeleton involvement in directional growth. Okay. Photoperiodism, phototropism is the growth of a plant towards light, while gravitropism is the growth in response to gravity. Cytoskeleton is involved in these processes by enabling the redistribution of organelles and molecules such as auxin, a key hormone that regulates directional growth. In phototropism, the cytoskeleton helps relocate auxin to the shaded side of plant, promoting cell elongation on that side and bending towards light. In gravitropism, the cytoskeleton facilitates the movement of starch-filled organelles in root cells that settle in responses to gravity. Their, re- their repositioning triggers cytoskeleton change the direction that direct oxygen transport and result in roots growing towards and shoot growing upwards. Uh, Cytoskeleton elements like actin filaments and microtubules coordinate these movements, ensuring proper growth orientation. Move on to the next slide. Now let us discuss the dynamic nature of cytoskeleton in division, which is essential for various cellular processes like movement, division, maintaining cell shape. So let's break it down. The first is rapid assembly and disassembly. Adapt to cellular needs. The cytoskeleton is not a static structure. It can quickly assemble and disassemble based on the needs of cell. For example, during cell division, microtubules rapidly form the mitotic spindle, and actin filaments can recognize to enable cell migration. This flexibility allows the cell to adapt the changing condition, regulation, the changing conditions. The next point is regulation by signaling pathways, calcium ions, kinases, and other molecules. The cytoskeleton's dynamics are tightly regulated by various signaling molecules. For instance, calcium and ions and protein kinases can influence the polymerization and depolymerization of cytoskeletal components. These signaling pathways allow cell to respond to external signals like hormones or changes in the environment and coordinates its internal processes like movement, contraction, and shape changes. The third point is interplay between microtubules and actin filaments. Microtubules and actin filaments are two primary components of cytoskeleton. They often work together to maintain cell shape, facilitate intercellular transport, and enabling cellular movement. Uh, for instance, in cell migration, actin filaments push the cell membrane forward, while microtubule help in stabilizing structure and positioning organelles. The coordination between these two structures is crucial for cell to function properly and respond to the various stimuli. Move on to the next slide. Uh, to summarize, the cytoskeleton is the central to plant cell structure and function. It supports intercellular transport and cell division. Also, it plays a vital role in environmental adaptation. The cytoskeleton's dynamics and its interconnected natures allow plant cells to respond to challenges and stimuli with remarkable precision. Understanding this network deepens our knowledge of plant biology and provides insights into how plants grow, develop, and adapt to their surroundings. Such knowledge not only facilitate, facilitating but also has practical applications in areas like agriculture and biotechnology. Thank you for your attention throughout this lecture. I hope now you have a deeper appreciation for cytoskeleton's complexity and its critical role in plant cells. If you have any question, I'd be happy to address them now. For further reading and or exploration on this topic, feel free to reach me out or refer the resources shared in the presentation. Thank you once again. I look forward to our next session.